For those who don't know me, I have started as faculty in electrical engineering last November. So it's been the best of times and it's been the worst of times because I'm trying to put a lab together, and we were like for 2 weeks in the lab and then we were sent back home. So I hope you're all doing as well as you can given the current situation, and you're all brave to be enduring like your studies during those crazy times. So yep. So again, ask questions. I think I have a little bit of a delay between my my internet's not super good, but let's start. So I like to call myself a quantum engineer. This means that I'm trained as an experimentalist and that I build apparatus to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that we learn in high school and that rule everything big around us. So I messed up. I did not have the time to change the title. This is a sort of quick, broad talk that I have to talk a little bit about my academic path. And I'm happy to talk about this academic path in more details and at some point soon. Right. So I'm not only I'm my background. So I'm from Brazil. I started my undergraduate in electrical engineering for 2 years. Then I got a scholarship to study in France, where I studied physics. I finished my undergraduate studies in physics, having started in electrical engineering, did a master in physics, then a PhD in electrical engineering, then a first postdoc in chemistry, a second postdoc in bioengineering, and now I'm faculty at UCLA in electrical engineering. And I hope you will agree that to some extent it makes some sense. Okay, so for most of my life, I was used to dealing with these sort of sensors that you can see in this slide there, right? But some years ago, I realized that nature very smartly surpasses humankind-made sensors in the craziest of ways. And I'm going to share you a little bit on how I got interested in living sensors in general, and how I put my training as a quantum engineer to study sensors in nature. But before I explain where I'm going, I need to tell you where I come from. And we're going to start talking a little bit about quantum mechanics. You don't need to have taken quantum mechanics, but I'll start talking about very technological things. And very quickly, we will have talked about things of relevance for biology, such as organismal migration, how our cells react to chemical stress, how cells interact with radiation. And I hope to be able to convince you and talk a little bit about how one can use technology, like how one can actually improve technology by thinking about ways that nature works. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at very, very tiny scales. So I'm going to open a parenthesis, and this parenthesis will be closed to tell you where where I'm coming from. And to motivate this parenthesis, I have to tell you that I think humankind is obsessed with measuring things better because it means that we understand nature better. So we can think about, for instance, measuring better frequencies. This is a so-called atomic clock, which is currently at NIST. And with this atomic clock, one can define in a very precise way what a second is based on an atomic transition. So we can think about measuring better frequencies or like defining the second better. We can think about measuring better magnetic fields. So this is the magnetic resonance imaging of a baby inside the mother's belly, and this is enabled by measuring tiny magnetic fields in the field of magnetic resonance. And one can think about measuring, for instance, better accelerations so that your gaming experience is enhanced. Such tiny accelerometers, they're ubiquitous in all sorts of like small iPhone, Android, and video game devices. But the question that I ask you is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity to be measured is very, very small? I'm going to argue that in this case, you need a tiny little sensor that is able to measure tiny little quantities. If the sensor is tiny, let's make it very, very tiny. Let's make it quantum. That is, let's make it an object that is well described by the laws of quantum mechanics that rule at very tiny length scales, at the scale of a couple of atoms, a couple of molecules. I won't have time to thoroughly explain this, but there's a whole body of mathematical arguments saying that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. I am going to very briefly describe two technological quantum sensors that I worked with in the past and how they're informing how I'm working with living sensors right now. So the first technological quantum sensor that I worked with was a single spin in the material, diamond, that we have in our jewelry. And one could put this very tiny spin in diamond to work as a quantum magnetic field detector. So physicists, can you see my props? Can you see my arrow? Hello?
Yeah, we can
Yes,
see.
we do.
Cool. Okay. And someone, could you please monitor the? Oh, if there are questions, please someone feel free to unmute yourselves because I won't be able to be monitoring the chat. But physicists and engineers, they depict the merely quantum property of spin, usually with a arrow. Spin is a property that has no equivalent in classical physics, and it represents how well a small quantum object, such as an electron, interacts with magnetic fields. So spin up, the arrow pointing up or the arrow pointing down, it only signifies two different energy levels of this merely quantum property called spin. So we can put a single spin in diamond to work as a magnetic field sensor. So this spin in diamond is actually arises in the form of a crystalline defect in the diamond lattice. And this crystal defect is actually a color center. A color center, so color centers in general are crystalline defects in the solid state lattice that explain the color of those diamonds that you see there in the bottom picture. Those color centers, they absorb light and then they emit light back. It's also called fluoresce. They fluoresce back, and that's why, and the fluorescence of each defect is a little bit different. So that's why we have all those different colors of the diamond. The diamond, the defect that I worked with, and that is a very sensitive magnetic field sensor, is called the nitrogen vacancy center. It arises naturally, but it can also be engineered, and it comes up when a missing carbon atom, a vacancy, it sits nearby to a nitrogen, which is one of the most commonly occurring substitutional impurities in the carbon lattice that makes diamond. So the cool thing is that when a vacancy sits next by to a nitrogen within the carbon lattice, it's sort of a mess of electrons, right? The vacancy leaves electrons unpaired. The nitrogen has a different number of available electrons than the carbons. But something almost magical happens, and that's the fact that once you calculate the possible quantum mechanical energy levels that that this defect, this atomic size defect can have, those quantum mechanical energy levels, they simplify a lot, and they actually are similar to the energy levels of one single electron, that is one single electron spin, so that we can look at the solid state complex and think about it in terms of like one energy represented by arrow up and one energy represented by arrow down. So the way that we find such defects in the lab is the following way. We put a diamond slab onto a moving state, and this diamond slab doesn't need to be anything special. And actually the very first diamond slab that we worked with had been given to us, like discarded by a jeweler. Okay, so you get your crappy diamond slab on top of a moving state, and you sort of scan it while you're shining a laser through this sample. As I mentioned, this defect is a color center, that is, it absorbs light and then it fluoresces, so that as you're scanning your diamond, the moment your laser, your light hits such a defect, this defect gets excited, absorbs light, and then emits light, fluoresces, and we can collect this fluorescence in the lab. This is what it looks like at increasing zooms. Okay? It's quite remarkable because in the bottom right picture there, you are actually looking at the fluorescence, the light signal of one single spin in diamond.
Hey, Clarice, I
Yes.
just had a quick question. So
Yes.
when you mix the 532 nanometer laser with the 637,
Yes.
does it create like and like beats or whatever, like a Gaussian wave packet?
So first of all, we use a laser, this green laser, just because it's convenient. It doesn't need to be a laser, and that's and this I mean for the same reason that color centers, they exist even with regular light, right? So it doesn't need to be a laser at all, but in the lab it's more efficient to excite those centers and more convenient to use a laser. And this laser is well represented by a Gaussian beam. Yes. Is this the question?
Oh, I was just wondering if like the wavelengths kind of like mix, like so there's like one wave with like 637 and another wave with like 532 nanometer wavelength.
Yeah. Oh, great. That's a very cool question. And this is the reason why we have this dichroic mirror here in this top right scheme there. A dichroic mirror is a mirror that reflects certain wavelengths and lets through certain wavelengths. So when green laser hits the sample, some of it is scattered back, but it does not go through our detector because this so-called dichroic mirror blocks it. It only leaves certain bands of light go through, and in this case, we leave the 637 nanometers and around it go through. And that's how we make the difference between the light that is exciting the diamond and the light that is being collected from the excitation of the color center.
Very cool.
It's very cool, right? But it actually gets cooler than that. And for reasons that I won't have time to explain, just by looking at how strongly this thing, this atomic-sized stuff in diamond is emitting light, we can actually infer whether the spin state, whether the energy levels, the quantum energy levels, are related to the arrow up or the arrow down. I'll repeat, just by looking at the fluorescence that is being emitted in your lab, you can actually make a measurement of the spin state of this defect, which is crazy if you think about, right? But and this is how we can like in a very simple way have access to the quantum properties of this thing.
Would you mind? I think I haven't taken quantum
Yes.
course at all. So would you mind explaining like what effect the spin has like on the amount, the quantity that you're measuring? Since I don't quite have like a clear connection from like how you're able to measure it from the effect.
Okay, so the premise of this work was to put a single spin to work as a very good quantum sensor for magnetic fields. I did not explain to you yet how it works, and I will.
Okay.
Right? But to cut a long story short, different spin states are going to react differently to different magnetic fields. Okay, and I didn't tell you how that works, but the important part is that you can actually read out the spin states via the intensity of light that this stuff is emitting. So we're going to get to the magnetometer part, but before that, I want to point out that just by looking at how strongly this thing is emitting light, you can actually have a measurement of quantum properties of this spin, and we're actually going to use this this measurement of light, this measurement of the spin state, to infer magnetic fields in a while. But it wouldn't be possible if this property wasn't there, if we didn't have a relatively easy way of reading out the spin states via fluorescence.
Okay.
We're going to get there. So again, actually we're going to get to it right now, right? Because we can put it to use, as I was just saying, as a very sensitive magnetometer. And the way that it works is the following. I just write there two different spin states. They could have been called anything, arrow up and arrow down, star and square, whatever. Here I'm just calling them 0 and 1. Those two spin states, they have different energies. And the difference in energy is on the microwave range, more or less in the band of what you have in your own kitchen. Right? And it turns out, and that's the key part for sensing magnetic fields, it turns out that one of these states, the one that I call 0 here, it doesn't care about external magnetic fields. Whereas the state that is called there +1, whenever it sees an external magnetic field, it actually gets promoted in energy. Its energy changes by a tiny little amount, delta. Okay? And this is actually very, very important, because delta, by something called Zeeman interaction, which is how spins interact with magnetic fields, this shift in energy is proportional to the magnitude of the field. And this should be of like interest to all of you electrical engineers, because, and this is a fundamental point that doesn't often get made, measuring a magnetic field is going to be mapped onto the problem of measuring a tiny detuning, a tiny shift delta from a known, what I call here resonance, from a known difference in energy. The idea is that in the absence of magnetic fields, we know exactly what the difference in energy between up and down, or here 0 and 1 is. But there's an extra field, so that what one is trying to do as one measures to infer the magnetic field is actually trying to measure this detuning, this little shift in energy that we have, basically two states have. And maybe some of you have taken some signal processing classes. Measuring shifts from a known energy difference is something that engineers have known how to do for like decades, right? And this is super important. This is how we can use spins to sense a magnetic field, but there's a catch. I won't be able to talk a lot about why that is, but everything that starts quantum dies classical, and that's sort of the reason why we live in a mostly classical world. What happens is that quantum states are very fragile, so that when quantum things start interacting with each other, they push them down to like become classical again as things get larger in space and hotter in temperature. So this way of measuring magnetic fields in a very sensitive way is limited in time, only while this spin is well described by the laws of quantum mechanics. And this property can now be engineered, but in a crappy diamond, the quantum properties last for about 2 microseconds at room temperature, and like in a big crystal. And that's like, for people who do this field, is called quantum sensing, or also like for people who do quantum sensing and quantum computing, 2 microseconds at room temperature is very, very long. So really the fields of quantum computing and quantum sensing, they're all concerned with ways of extending the quantumness of systems so that you can play with quantum objects for longer and longer. Think of it as a signal processing problem. The longer you're able to acquire a signal, when you Fourier transform it, the better the frequency resolution that you're going to have. In other words, if you extend the quantumness, you reduce the noise in your system. And this has been known for many decades too. One way to extend quantumness and reducing the noise is by applying tailored electromagnetic fields. In this case, you would apply tailored microwave fields that have very particular frequencies, phase, and sometimes amplitude. And there's people who actually make a living out of devising better electromagnetic field sequences or pulses that extend the quantumness of tiny objects. What are your questions right now? So and feel free to interrupt, but the big idea here is then to have a tiny little sensor that you can bring in close proximity to a tiny little sample, right? So that you could infer the magnetic field generated by this tiny little sample. And this is how one put a single spinning diamond to use as a tiny and very sensitive magnetometer.
Also, I had a question.
Yes.
So I was wondering if the sample had any behavior similar to an antenna or waveguide.
So here's what the sample does. The sample produces magnetic fields, and one wants to measure those magnetic fields, right? So in this sense, I mean that's you don't even need to think about the sample as an antenna or something. Effectively, it is, if you will, producing an effect on your sensor, and this effect, for instance, spins in the sample like little arrows from other electrons or from spins related to the nuclei of atoms, they are actually producing tiny magnetic fields, and those magnetic fields can be sensed by your tiny little sensor. What one could do is actually, there are ways of enhancing both the signal produced by the sample, and there are ways of enhancing the sensitivity of your single spin in diamond. But for all purposes, your sample is just something that creates a magnetic field, or you don't even need to have like the goal of the sensor is that it can sense tiny little magnetic fields, say produced by one or a couple of atoms. But imagine that the thing that produces the magnetic field could be anything, could be like a tiny coil or whatever. For instance, nitrogen vacancy centers are starting to be used right now to help in the detection of oil when people do they look for deposits of petroleum or something, because those defects in diamond, they can be sensitive to tiny magnetic fields produced by certain spins in organic matter, in carbons, basically. And so your sample can be anything that produces a magnetic field. The good thing about these sensors is that they work at room temperature, right? So that you can bring things to the field, you can do experiments having to do with biology, and they're very sensitive. You don't need to have a huge quantity of sample for them to work or to pick up signals.
Gotcha. So it doesn't have to be like super cold, like superconductors and or stuff.
No. That's the whole. I didn't mention it, but I think I'll make a point in a couple of slides later on. But this works at room temperature and under, well, the messy conditions of a diamond lattice, right? This is a sensor that exists in the solid state, and it picks up signals from messy samples. And this is going to be super important for things that I'm going to talk about in a couple of slides. Cool. Okay. So I think I have some slides on so I've just talked to you how I worked with one technological quantum sensor, this spin diamond. I think I have some slides on work that I did on a technological quantum sensor that that is related to imaging. Let me see if I have some slides. Oh no, I don't. But if we have time, I'll be happy to talk more about that. But magnetic field sensors are not the only technological quantum sensors that exist, right? You can, with quantum sensors, you can measure temperature, electromagnetic fields, you can do imaging. And I also worked with this other technological quantum sensor that helped with imaging. And in particular, what those two sensors have in common, I only described the left one, but those two types of sensors they have in common is that they're quantum. They work at room temperature and under noisy environments. Okay, that they can pick up signals, especially from like disordered materials, things in the solid state at room temperature, biological things. So just for you to have seen it explicitly mentioned that quantum sensors at room temperature, they exist and they function. What are your questions before I close the parentheses that I opened a couple of minutes ago? if you don't have questions now, I hope you will have questions in what's going to follow, because I'm going to make the leap now from those technological quantum sensors into what I'm calling living quantum sensors. Okay, and the idea is that I'm going to mention now a naturally occurring magnetic field sensor that is in many ways, that works in many ways very similarly to the technological quantum sensor that I just described to you with the spin in diamond. But in order to talk about that, to talk about quantum in biological systems, I need you electrical engineers to humor me. So because I need to talk about chemistry, like biology at the nanoscale, which is basically the chemistry. Okay. So, and this is all things that I've been picking up in my. So I am not trained as a biologist or chemist, but I hope I can convince you that there are some things happen in chemistry that have tremendous technological implications. And let me see if I can convince you like that it's cool too. So it's been known for many decades now in a field called radical chemistry, from not radical as opposed to conservative, radical in the sense of radical species, which are basically free electrons in reactions. This field of chemistry knows, it explains that magnetic fields can alter the final products of chemical reactions, okay, if these chemical reactions have to some extent impaired electron spins. Okay, I'm not going to give you all the details, but this is the important idea. There are some chemical reactions that depend on spin, and I hope you can see my arrow again, in that they continue through one chemical branch if the spin is up, and they continue through another branch if the spin is down. Importantly, the final products of those chemical reactions are macroscopically different. So a spin, like a spin state happening at the very top of this chemical reaction can actually influence macroscopic pathways, giving rise to very different final products. And as I mentioned a little bit before, which is exactly the same with the spinning diamond, magnetic fields influence spin states differently. That is, magnetic fields might not care, state up may not care about magnetic fields, and state down might care about magnetic fields, so that a small magnetic field can actually alter the probability of the spin being up or down, so that it alters the probability of the chemical reaction going through one pathway or the other pathway. So this is known, okay? So don't look at the top left picture. I can explain it later, and for those of you who have taken quantum mechanics, but this effect has been demonstrated at room temperature, in solution, in the gas phase, in the solid state, and for magnetic fields as tiny as the magnetic field of the Earth, which is about 50 microtesla, and which is smaller than the magnetic field that you feel when you hold your cell phone close to your ear. Okay. And right now, there is no other way to explain this that does not go through this quantum property of spin being actually altered by magnetic fields. I would like you to look at this bottom left figure in which I'm going to try to explain with an analogy how a very fragile quantum property of spin might actually make a difference, like big time, macroscopically in chemical reactions. So if you want to engage the help of a fly to tip a big rock, right, to one side or the other, if the rock starts on its thermodynamical equilibrium state, like in one of its flat edges, the fly landing on the right or on the left, it's not going to help the rock tip one way or the other. However, if this rock instead starts at a non-thermodynamical equilibrium state, say on its edge, right, the fly landing to the right or to the left actually might make a difference if the rock tips to one side or the other. In this analogy, the rock starting at its tip signifies a quantum mechanical state. As I mentioned, everything that starts quantum dies classical. And another way of looking at this is that quantum mechanics only exists as long as the laws of thermodynamics have not kicked in yet. So this is why the tiny help of a very fragile spin up or spin down, if we start in a quantum state, might actually help macroscopically one thing to get done or the other. What are your questions right now? Because this is a lot to absorb, and I'm not sure if many of you had thought about quantum mechanics being important for chemistry. I know that I got a whole new appreciation for quantum mechanics, for actually for chemistry, after I understood how much it was important.
Right,
Questions?
Yeah, so yeah, I had
Oh, hello, Joey. Hello.
Hi, Clarice. Yeah.
Yes.
So I kind of had like a clarification kind of on this, like this analogy.
Yes.
So is there basically like there's multiple ways to get from the reactant to the product, and like could be like a hard way and like an easier way, and so by putting in this like energy, you like influence the reactant to go to the product in like the easier way.
So I skipped a lot of stuff in this explanation, and one of the things that I skipped was the fact that usually the type of chemical reaction that depends on spin, they are photodependent. That is, they start with the absorption of a photon. Okay, and this photon actually gives enough energy in this analogy for the rock to tip over. So we can talk much more because you know a lot of the details of this model, but I glossed over it. But at some point, there is actually, you start with the rock on its flat edge. There's a kick in energy that is a very macroscopic kick. It's a lot of energy involved, and it's a classical sort of jump in energy. And from there on, this tiny effect can play a role, but not before that. So in all that I'm saying, if you didn't have this energy kick provided by the absorption of a photon, you wouldn't give a chance for the quantum mechanical effects to play a role, actually.
Right. Okay, yeah, thank you.
But we can talk about that later. And I glossed over the fact that most reactions that are spin dependent, they are kickstarted by the absorption of a photon. This means that they are photodependent chemical reactions.
Mm-hmm. Right. All right, yeah, thank you.
More questions, people?
I guess
So
just to sort of make sure I
yes,
understand. I
yes,
guess
go for it.
just to make sure I understand like what's going on in this diagram here. Essentially, what's happening is a photon is providing its energy to some quantum system where that's putting it into a state where where the spin of individual elements, which normally wouldn't have any sizable effect, but in this sort of like threshold state almost, the radical pair, in that exact state, a single spin has enough effect to cause the state to converge into either one or the other
Yes.
final state.
I could not have put it better. Yes.
Okay, cool. Okay.
Okay. Which is super cool. And I'm glossing over the fact that it's not only one spin that is involved, it's actually two spins, but that's not important. What is important is exactly as you said, we get kickstarted by a photon. This photon gives enough energy to put things in an out of equilibrium state in which tiny little quantum tweaks, such as the different energies of an electronic spin, can actually play a macroscopic role. Yes. So since the end of the 70s, actually 40 years ago, biophysicists, they wanted to sort of understand, and this is like the historical side of it. Right now, we know that what I'm about to tell you, it's way more widespread than that, but this whole shebang that I'm about to explain to you started because biophysicists, they wanted to understand how birds migrate. Okay, it's known without any doubt, actually, that at least as a partial cue, birds, when they migrate north to south, they use the magnetic field of the Earth, which is, as I said, super, super tiny. Like, so how do they do this? So 40 years ago, theorists posited, there were no experiments yet, but they posited that were the same sort of chemical reaction that depends on spin, were those to happen like within an organism, right? And pay attention at that. An organism could sense magnetic fields to the extent that they could sense different physiological concentrations of final products coming from these spin-dependent chemical reactions. I'll repeat that. Birds do use the magnetic field of the Earth for something at some point in their journey. And it was never understood why. So people started hypothesizing that if spin-dependent chemical reactions that depend on spin-dependent chemical reactions were to be happening like in the wet, noisy environment of a cell, organisms could sense magnetic fields because they could sense different macroscopic final products of these chemical reactions. And it was just a hypothesis. And at that point, I didn't, so as I just mentioned, because of Joey's question, usually chemical reactions that depend on spin, they're photodependent. That is, they are kickstarted by the absorption of a photon. So biophysicists, they started looking for things in organisms that absorb photons. And I don't think it comes as a big surprise that they started looking for things in like the eyes of organisms. So at that point, like decades ago, the only animal photoreceptor, that is the molecule that absorbs light in animals, the only animal photoreceptor known to be able to sustain spin-dependent chemical reactions was a protein called Cryptochrome. Okay, right now it's no longer true. There are many other animal photoreceptors that can sustain spin-dependent chemical reactions, but Cryptochrome was the only one at that point that fit the bill. Cryptochrome, again, is a photoreceptor that is present in the eyes of migrating birds, in our eyes, in the antennas of migrating butterflies, but it's also present in all, I'm going to repeat this, it is also present in all our cells because it has circadian rhythm regulation functions. That is, it regulates when you go to sleep, when you wake up, how you interact with light. For instance, you might sometime have traveled and crossed time zones. When you're jet lagged and people tell you to look at blue light, this is not quack science. This is, I mean, people got the Nobel Prize for that. The reason why blue light might help with jet lag is because it sort of interacts with photoreceptors such as cryptochrome in your eye. So cryptochrome is present throughout the tree of life. All of those organisms there have been shown to express cryptochrome, to have cryptochrome in their cells. But there have been some studies that actually are consistent with magnetosensing in organisms being related to cryptochrome. And those the organisms to which there are such studies, they are circled, they are squared there in red. And yes, if you see, humans are right there to the left of the whatever crab or lobster. The problem is that evidence for cryptochrome-based magnetosensing is really, really widespread, but unfortunately at very disconnected length scales. So there's evidence for magnetosensing in cryptochrome in chemistry for like vials of proteins, and there is evidence at the level of many, many organisms. Let me talk to you about the very tiny length scales, the chemical length scales. So in the top left picture there, you can see what happens when you excite vials of cryptochrome with a laser. Okay, experimenters took the curve of the fluorescence of cryptochrome in solution as a function of the time of excitation with a laser. The first thing that you see is that this fluorescence is dramatically going down. This means that the pigments are being killed by your laser. But as the researchers pulsed a magnetic field on and off during the excitation with the laser, you could see that the fluorescence sort of was modulated up and down. In a way, I'm not showing the magnetic field profile, but the magnetic field profile actually follows a lot what you see in the inset there. That is, the mean fluorescence of this pigment, of this photoreceptor, is modulated with magnetic fields on and off. And if you remember, this is exactly the thing that was happening with the spin in diamond. Just by looking at how strongly that the blob in your lab coming from the spin in diamond, how strongly that blob was emitting light, we could infer if the spin was up or down. And here it's actually the exact same thing. Just by looking at how strongly this protein is emitting light, we can actually infer if the chemical reaction went through the branch of spin up or the chemical reaction went through the branch of spin down. This is not to say organisms might be feeling their own fluorescence, intensity of light to sense magnetic fields, but this is a very convenient knob that we have in the lab. Okay, we can look at fluorescence intensities in the lab and we can infer quantum things about it. Another thing, this protein in solution has been shown to be quantum, to remain quantum for about 1 microsecond at room temperature, which is on the same ballpark as the 2 microseconds that I cited for naturally occurring diamond, the time during which the spins, that those particular spins in diamond were well described by the laws of quantum mechanics. So this is very encouraging. chemically, we have no doubt that cryptochrome is a magnetic field sensor. On the other side of the length scales, there are experiments done with like whole birds, whole flies that are consistent with things like with this spin hypothesis. Let me describe some of the evidence. For instance, apparently, and I have no idea how people do this, flies can be trained. And they can be trained in particular to find food conditioned on the presence of a magnetic field. So the flies learn that a magnetic field means there's food. And then researchers knocked out or removed the cryptochrome gene from the flies, and the flies are no longer able to find food based on the presence of a magnetic field. In a further study, the researchers put back onto the flies human, not fly, cryptochrome, and the flies are back to being able to find magnetic fields. Again, consistent with magnetosensing being dependent on cryptochrome. Birds, during migrating season, birds can be put, and that's the problem with experiments with birds, right? You have an N of like 23. You do experiments on 23 birds, which is like 3 months of research, but that's very tiny for like engineers, right? You do experiments with 23 birds. You put them in cages, you apply different magnetic fields, and you see that during migration season, depending on the magnetic field that they see, they want to go out of the cage following different directions. A cool experiment that people did with birds was the following. The assumption is that there is a spin that feels the magnetic field of the Earth, right? And that helps birds navigate by driving a chemical reaction. The magnetic field of the Earth is a DC magnetic field, which is of 50 microtesla, as I mentioned. There is something that you might learn in your advanced quantum mechanics course that is called Larmor Frequency. This means that a spin will process like a little top around a magnetic field with a frequency that depends on the field strength. So this is called the Larmor frequency, and the Larmor frequency of the magnetic field of the Earth is about 1.4 megahertz. So the idea is that the magnetic field of the Earth is influencing hard a chemical reaction. But imagine what would happen if you put a tiny magnetic field onto this mechanism that were to alternate, right, be AC with the frequency at which the spin is processing. If this model were true, what would happen is that you would tap onto a resonance, thereby making this spin like process like crazy. And actually, you can show that it would wash out the effect of the magnetic field. So magnetic field of the Earth, 50 microtesla. Researchers put, I think, 200 nanotesla at a AC frequency matching this Larmor frequency of 1.4 megahertz, and they saw that the birds got disoriented. That is, they didn't have a preferential way to go out of the cage. So all of this is very cool, but it's very hard to say that flies and birds, they are behaving the way they are because they're quantum, right? There's quantum things that are relevant for their physiology. So those length scales are very disconnected. So that's exactly what we're trying to do in our new group at UCLA. We want to bridge those length scales. Basically, what we want to do is start doing chemically inspired experiments inside, say, one cell, a couple of cells, and to cut a long story short, we just want to be doing experiments under a microscope. We are going to be treating spins in proteins inside cells as bona fide quantum sensors in the same way that we treated the spin in diamond. This just means that we're going to have tiny coils, a microscope, and we will be looking at the effect of magnetic fields onto proteins in tiny living systems, right? To be able to see if we can step by step either establish or refute that quantum mechanics might actually play a role for physiological things. Again, we want to try to see, as we saw it was possible with technological quantum sensors, if living things could be quantum sensors working at room temperature and under very noisy environments. The cool thing about all of this, and so again, I'm trained as an engineer, as a physicist, right? I have always hated biology and chemistry, but the more one starts reading about it, it's like, if this is true, the implications of like spin dependent bio things is like crazy. For instance, this very same spin mechanism that I sort of quickly explained. So there are results consistent with the same sort of mechanism being behind metabolic regulation, for instance, or like DNA regulation, growth growth and regeneration of cells. Let me just describe one experiment. So bio people have these like black boxes that they call, I think, metabolic analyzers. They're just like an apparatus where you put a plate of cells inside, and then you do some gas reading of byproducts of metabolic phenomena, like how much like respiration stuff you have in the cells at the level of like thousands of cells.
Yeah, I have a
Physicists,
quick
they
question.
yes,
Yeah.
yes.
So what's the difference between magnetic spin and like electron spin? So I was a
Sorry,
little confused.
sorry. This is so electrons and other quantum objects have spin. Spin always has to do with how quantum objects interact with magnetic fields. So the only type of spins that exist are magnetic spins, like there is spins that interact with magnetic fields, but electrons, the elementary particle, they have spin. So it's always, the spin always has to do with interactions with magnetic fields. But one type of quantum object that has spin is an electron. Other types of objects that have spin are like some nuclei of atoms. Right? So when I say, I don't know if you read electronic spin. So electronic spin means the spin of the electron. But spins, the quantum property of spin always has to do with how the quantum mechanical object interacts with magnetic fields. Sorry, it's I had never thought about this, but this is confusing, indeed. Cool.
Yeah, gotcha.
Okay, I'll be more mindful next time. But again, electronic spin here is just like spin of the electron, and spin always has to do with the interaction of the quantum object with magnetic fields.
Thank you so much. Yeah.
So back to the black box. Physicists they shielded this machine against external magnetic fields. They put a plate of cells inside, and they applied inside tiny magnetic fields that changed in frequency, in direction. And what they saw was a macroscopic difference in metabolic parameters, again, such as respiration rates of the cells, the pH of cells, having to do with differences in very tiny magnetic field directions and frequencies in a way that is consistent with this model that I very simply described. Again, it's a lot of stuff. There is a lot of like evidence that this might be relevant for biological phenomena. So, and I hope you might get a kick out of this, but there's a lot of technological phenomena that are also driven by spin phenomena. And in many ways, when you think about transport of particles inside a cell, this is like inside a biological cell, it's not very different from transport of electrons, say, in a solar cell, right? When you need to have electrons go one way and holes go the other way. There's a lot of technological principles of technologies that are spin dependent. And all I'm saying is that maybe we can learn with nature, right? Because if nature really is using quantum mechanical things to function better, it has been optimized over millions of years, right? And nature really knows how to deal with noise. So I think there's a lot of things that we could learn from nature. And on top of that, right? If quantum makes a difference for biology, can we start harnessing the quantum mechanical degrees of freedom to like drive physiological outcomes? Can we play with spins and other quantum objects inside, say, biological cells, in the same way that people play with quantum bits for quantum computing, right? With the goal of actually driving metabolism, driving healthcare in a way that is like using quantum mechanical degrees of freedom. So those are the two main things that we're trying to do in our lab. Learn with nature and commandeer nature via quantum degrees of freedom. And for instance, and there's some like crazy implications. For instance, space travel. Okay. A group of physicists whom I know and who are like, who do what's so-called precision measurements. So their life is measuring tiny quantities. They can build like cages called hypomagnetic cages, whose like noise level is absolutely like less than a nanotesla or something. And inside those cages, you can then apply very controlled magnetic fields for kicks. And this experiment has been repeated twice, but both in physics labs. So the bio people, they don't trust those results very much, but I do. So those physics people, they raised tadpole, like frog thingies, like the thing that will become frogs, inside those hypomagnetic chambers, once with an applied magnetic field that mimicked the magnetic field of the Earth, and another time without any magnetic field, that is only with like a nanotesla residual field. And what they saw was that in the absence of magnetic fields, like nanotesla level, 30% of the frog embryos of the tadpoles were malformed. Okay, which doesn't mean it has anything to do with spins or is quantum, but it means that there's some very sensitive tuning and interacting with the magnetic field of the Earth that might be doing things for the frog embryo. So what happens in space travel, right? What's the magnetic field that astronauts will be exposed to when they undertake like 20 years travel to whatever, to another planet, or Mars, right? What's the magnetic field on Mars? Can we grow lettuce in Mars, right? So those are things that I think we can learn a lot, and especially like using the techniques of quantum mechanics to look at biological things. And someone posted like the levitating frog. What's that about, Koji?
Oh, it's just a frog being suspended in midair by an electric field or magnetic field.
Well, but that's different.
Oh.
I have no idea how that's done, but that's different. Here we're talking about this magnetic field is probably very large. What I'm talking about here is the presence or absence of a magnetic field that is smaller than the magnetic field that you feel from your cell phone. that it messes up like tiny development at the cellular level. And so this, all of this is super cool, and we think we are well equipped like to approach all of this in a very technological way. And this is what our lab wants to do. We want to either establish or refute that quantum physics play a role in biology, and importantly, if it can be manipulated to technological advance and like healthcare and therapeutic advance. And we're not the only people doing this. There's other groups doing this as well. For instance, there's something in quantum computing called photon entanglement. You might have heard or not about it. It doesn't really matter, but some people have just produced entangled photons from a protein, and there's other people doing things that you might have heard of, like teleportation, quantum teleportation in like biological molecules. So it's so cool. There's a lot of room, we think, for high-tech exploration of biology. If you're curious about this, that is called quantum biology. You might have a look at this book called Life on the Edge. Don't tell anyone. And like I didn't say this, but I have a PDF of that. If you're interested, I can share. It's a very good survey of quantum biology. And have a look at also this article that I whose URL I shortened there for you. So I really think that big science challenges are going to be unveiled via interdisciplinary stuff, right? So it's all a matter of being comfortable with being uncomfortable, because when I cannot solve this alone, the bio people cannot solve this alone, the engineers cannot solve it alone. So we're all doing things together, and most of the time is like an exercise in communication. I need to communicate with my chemistry colleagues and so forth what they mean, and right. And but I really have fun doing interdisciplinary research, even though many times I don't get 50% of what the bio people are telling me, but that's fine. I'm learning a lot. So we have just started. Write me if you're interested in talking more. We have some opportunities for internships. We are growing. We're limited in our growth right now by COVID, but we're building stuff, starting to build stuff in the lab. So if you're interested, let us know. There's a lot of optics building involved, and electronics and programming, and also there's room for simulations. And I don't know, Joey had a pretty hardcore. I don't know if Joey is still there, but he had a pretty hardcore introduction to quantum mechanics simulations, right, Joey? If he's there during this.
Yeah,
I don't know.
yeah. I still don't understand like a lot of it. But yeah, it was definitely interesting.
Joey, tell us like the first paper. Do you remember the title of the first paper that you read for your internship during the summer?
Oh,
do you remember the title? Oh my God.
Yeah, I can't remember off the top of my head, but it was definitely like using quantum machine learning for quantum neural networks for like quantum something. So yeah, definitely.
Yeah. So Joey was working with a very talented, mathematically inclined graduate student. So yes, but because we couldn't have anyone in the lab, so but yes, and then talk to Joey, and yeah, and that's all I wanted to tell you. And what are your questions? And before anything, may the quantum be with you. Come on, there must be a lot of questions. It's super exciting. What do you guys think?
So basically, you're just trying to show that every biological process is at the core some quantum process.
Well, so we want either to unambiguously establish or refute that, right? So if you go down to like each atom, each molecule, everything is quantum. But that's like trivially quantum. It's quantum because it's very small. What we're trying to either prove or establish or refute is whether quantum effects, like transient quantum effects, can have a macroscopic influence in the same way that it's known in chemistry that tiny spin effects can have macroscopic final products. And it's very hard, right? If it's very hard to make the jump from birds to spins in molecules, right? Because you don't know what the hell is going on with the bird. So this is why we want to be in the middle, right? To start doing very controlled experiments with like a single cell or some molecules in order to see how things work. For example, spin stuff, it's way faster than metabolic processes. For instance, spin dynamics, the dynamics of a magnetic field, as I mentioned like Larmor frequency of 1 megahertz, right? The spin processing is like megahertz or even like gigahertz fast, whereas metabolic processes, they're like millisecond long or second long. So one of our experiments is going to be doing exactly that. We're going to try to correlate very fast time scales of spin processes and measure spins and correlate that with much longer time scales like of metabolic processes that might or might not be affected by us trying to control the fast spin processes.
Very interesting. Thank you for your talk.
Would that alter that testing that you're doing for that? Would that involve like modifying some magnetic field to alter, or at least to measure or possibly alter like the frequency of like the quantum oscillation and measure the effect that has?
That's exactly what it is.
Okay.
So this spin effect, for reasons that I won't have time to explain, this spin effect, according to the model, right, is sensitive to differences in field intensity, frequency, and direction. So what we want is to raster in a systematic way field direction, intensity, and frequency. For instance, we have one experiment that is better to do things at different orientations for different field orientations. Another experiment that is going to be focusing more on different frequencies to tap at different spin resonances. And magnetic field manipulation is actually what's done in the field of quantum sensing. I mentioned in one of my slides that people want to play with like the spins in diamond for longer so that they apply tailored electromagnetic fields. So here we want to do the same thing. We want to apply tailored magnetic fields so that we can putatively play with this spin in biology for longer so that we can learn about it more and more. So which means that technically this means that we want to do a quantum sensing experiment in biological samples. It's really like what people in quantum sensing are doing, but our sample prefers to live under a microscope, right? But it's exactly quantum sensing in biology.
So what I was interested in is like, because you said the quantum effects in diamond happen at room temperature. So I was wondering if you could like build a large scale quantum computer just using diamond as the quantum processing unit, you know?
So, in theory, yes, and I talked a little bit about it in another talk. Wait, where is that? Okay, so let me try to go to the source. Oh, it stopped sharing. Just a second. So let me show you one. So the answer is qubit. Okay. Yep. And why I need a new computer because my computer is okay. Cool. Can you see my screen?
Mm-hmm.
Yes. Okay. So I showed you a picture of one spinning diamond that was called the nitrogen vacancy center, right? This is just the fluorescence of that one single spin, which is basically just a quantum bit, a qubit, right? And the idea to do a quantum computer based on diamond is really just to scale this up and to have arrays of defects in diamond positioned like in an orderly way, as the one that you see in the top right picture there. The cool thing is that those spin defects, they occur in crappy diamond, but they can also be produced in the lab. Basically, you have to irradiate your diamond and then apply heat so that things shift around. But if you do this properly, you can place those nitrogen vacancy centers in, say, a lattice, just like the one that you see here. And the idea is to make nitrogen vacancy centers communicate with one another and use other spins nearby each nitrogen vacancy center as local memory units. So this is how a potential quantum computer with diamonds would work. This said, this would be a good quantum computer because it works at room temperature. But right now, it performs much worse than, say, Google's super nice cryogenic quantum computer with superconducting qubits, right? It may be that one day, yeah, one day if you want to have your like quantum laptop, one might want to have a room temperature quantum computer, but right now cryogenic quantum bits work much better, just because if you cool things down, you actually decrease the effect of like thermal motions and quantum objects kicking one another and pushing them out of a proper quantum mechanic state. And this is why like superconducting qubits and things that get cooled, they perform right now much better than things at room temperature.
That makes sense. Yeah. Thank you.